So we just got done talking about virus structure, and we ended up with talking about bacteriophages, which are also called phages. And remember that those are the ones that just affect bacteria. So they're the viruses that infect only bacteria. We're going to talk about replication of different types of viruses. And the first stuff we're going to talk about is how bacteriophage replication occurs, and then we're going to spend a little bit of time talking about the differences between replication in viruses that affect animals. So before we actually get into the nitty-gritty of that, let's talk about some more general stuff. As I mentioned in the last video, viruses require host cells for reproduction or replication. Because of that, they are called obligate intracellular parasites. Obligate means they absolutely require it. They can't do replication without this. Intracellular means that they enter the cell in order to replicate, and parasites mean that they gain something from being inside that cell, and the cell is harmed. So because they can only replicate inside a host cell and they typically damage the cell, that makes them obligate intracellular parasites. Each virus has its own host range, which varies a lot. Some viruses are very, very specific, and they can only infect a certain type of cell within a certain species of host. Others may have only one species that they infect, but they can potentially infect a variety of different types of cells within that host species, and still others have are able to infect multiple different species, although that is a lot more rare. So the first step is that the virus has to enter the host cell in some way, and there's a lot of different ways that they can do this, but essentially the cell, the host cell, has to somehow recognize the virus as something non-foreign or something that should be allowed to enter the cell. And so the virus can have multiple different techniques of sort of making a lock and key fit on the outside of the host cell. So something about this virus is going to connect to a receptor on the host cell, which will essentially open the gates so that the virus can enter it. Once inside, the virus often, not in every case, but often destroys the DNA of the host cell and then basically tells the host cell to replicate its own DNA and its own proteins. So it uses the enzymes, ribosomes, ATP, etc., all of this machinery that's already inside the host cell, and it commandeers it to make new viruses. And then once the different parts of the viruses have been created by the host cell, so here you see that the host cell has made some capsid proteins or capsomeres. Here's the DNA of the host of the virus. The host cell then transcribes it into mRNA and translates it into more capsid proteins. So that's the host cell doing that. Also, it does it uses replication, and it creates more viral DNA. So once the viral DNA and the viral proteins are created by the host cell, these things can automatically self-assemble, and then these viruses can exit the cell, often by destroying it, and can go off and infect new cells. So here's another example of that, a demonstration.
A virus is an intracellular parasite that can reproduce only by taking over a host cell. A virus consists of a nucleic acid genome enclosed in a protein shell called a capsid. In the virus shown here, the genome consists of DNA, but some viruses have RNA. Some viruses are also covered by a membranous envelope that is derived from the membrane of the host cell. There is usually a lock and key fit between the proteins of the capsid and receptors on a particular type of host cell. The virus attaches to a host cell and viral DNA enters the cell. The viral DNA uses nucleotides and enzymes of the host cell to replicate itself. The viral DNA then commandeers other host cell materials and machinery to transcribe its genes into messenger RNA and translate the RNA message into capsid proteins. Viral DNA and capsid proteins then assemble into new viruses. Mature viruses leave the host cell, often destroying the cell in the process. The viruses can go on to infect other cells, spreading the viral infection.
Okay, so that was sort of a general understanding or a general description of how viruses replicate inside a host cell. So now we're going to go to some more specific stuff about the replicative cycles of phages. Remember, phages are the ones that infect only bacterial cells. We know that we understand their replication the best. They are the most studied, so that's why we're going to spend some time on those. Phages have two different reproductive mechanisms or replicative mechanisms, the lytic cycle and the lysogenic cycle, which we're going to spend some time going over. The lytic cycle is basically what it does is the virus infects the host, and then a bunch of new viruses are created, and then when the new viruses break out of the host cell, the host cell breaks apart or lyses, and the host cell is killed when that happens. So that is called the lytic cycle because of the lysis of the host cell. So again, the lytic cycle culminates in the death of the host, and that occurs when all the new viruses break through the host cell. A phage that reproduces only by the lytic cycle is called a virulent phage. So in this case, these phages only use the lytic cycle for replication. And this is what that kind of looks like. Here you have the bacteriophage, the genome's inside the capsid, the head part of the capsid. Here's the tail and the tail fiber. The tail fibers have these little receptor binding sites on the ends, and here are the receptors on the host cell, the bacterial host, and these match up to form that lock and key that we were just talking about. Once that happens, the phage can attach to the host cell, the bacteria. Here we see the circular DNA of the bacteria, that's the host bacteria. And once it's attached, the head of the bacteriophage compresses and injects the DNA into the bacterial host. So let's go through the stages of the lytic cycle. Here we have attachment. Here we have injection of the bacteriophage's DNA into the host cell, and that basically causes degradation of the host DNA. Then the DNA that's been injected in there basically shuts down all the function of the bacteria and uses a bunch of materials and mechanisms to make the host cell perform transcription and translation replication of the bacterial DNA and of the bacterial proteins. And then once those are formed, the bacteria doesn't need to do anything else because the phages, the viruses, self-assemble within the bacterial cell. And once they're all assembled, then the viruses break through the cell membrane and destroy the cell through lysis. And they are released, and then they go on to infect other bacteria. So here's another description of that.
Bacteriophages, or phages, are viruses that infect bacteria. This is a T4 phage, which consists of DNA inside a protein coat.
Sorry about that.
The lytic cycle begins when the tail fibers of the phage stick to receptor sites on the surface of a host bacterium, such as E. coli. The phage injects its DNA into the host cell, leaving the empty protein coat outside. The DNA of the host cell is destroyed, and host cell enzymes and nucleotides are commandeered to replicate the phage DNA, making more phage DNA. The host cell's enzymes and ribosomes transcribe the phage genes and translate them into phage proteins. Phage parts accumulate and assemble to form phages. A phage enzyme digests the bacterial cell wall and the cell ruptures or lyses. As many as 200 phages spill out. Each of them may go on to infect another cell. This diagram summarizes the lytic cycle of bacteriophage T4.
Okay, so you can't really see that figure, but you've seen it, and you've seen the demonstration. You've also seen the figure before, so hopefully that's okay. So the other cycle that can happen is a lysogenic cycle, and this also replicates the genome of the phage, but in this case, it doesn't destroy the host right away. The lytic cycle has to be started in order for the bacterial cell to be destroyed. So in this case, the viral DNA is incorporated into the host cell's chromosome, and every time the host cell reproduces, it creates another copy of the bacterial DNA. So you'll see how this works in just a second. Some phages use both the lytic and lysogenic cycles, and those are called temperate phages. Okay, so here's how the two of them work together. This is the one we just talked about, the lytic cycle, where essentially, and this is a different type of phage. This is called the lambda phage, but it's still a bacteriophage. So in this case, we're seeing the lytic cycle where the virus DNA gets into the bacteria. The bacteria ends up making proteins and DNA for the virus, and they self-assemble, and then the cell lyses. Okay, so we understand the lytic cycle already. Here is the lysogenic cycle over here. We have, actually, so over here we have, this is right after the viral DNA has entered the bacteria. The lysogenic cycle is over on this side, and so you see that the viral DNA gets incorporated into the bacterial chromosome here, the circular chromosome. Once that happens, it's called a prophage. The prophage is copied every time the bacteria reproduces, and so you can see that here's a copy, here's the sister cell, and it also has the prophage in it, and it reproduces over and over and over again, and so the prophage continues to be reproducing with the bacteria. Okay, so this cycle can occur multiple times before something happens, and it can enter the lytic cycle, in which case something changes, and instead of incorporating into the bacterial DNA, it dissolves the bacterial DNA and commandeers the machinery of the bacteria to create more of the viruses, and then the lytic cycle happens, or lysis happens, releasing hundreds of phages. Okay, so again, here is the lytic cycle, which we've just talked about. It's just up close picture, a little bit better. Here's the lysogenic cycle. And again, this is just what we saw, just a little bit larger. So here's where the bacteria incorporates into the DNA. Here's where the virus incorporates into the bacterial DNA, and I'm not sure if that's what I said just a second ago. Here the bacteria is reproducing, and the prophage, which is the viral DNA, gets replicated as well. Every single time the bacteria divides, it's making new virus. And then eventually this will, if we're talking about a temperate virus, this bacteria and virus will enter the lytic stage at some point. So we've already talked about this. It's the prophage is the viral DNA that's integrated into the bacterial DNA. Every time the host divides, it copies the phage DNA. We didn't talk about this yet. So what causes the change from the lysogenic cycle to the lytic cycle? Some environmental signal. It really depends on what virus we're talking about, but it can be a chemical or a hormone or radiation can change the lysogenic cycle into the lytic cycle, which is where you have much more rapid replication of the virus and much more damage, because in that case, remember, the lysis is occurring and it's destroying the host cells. But again, we're still talking about bacteria here. Okay, here's an animation demonstrating this whole thing.
In contrast to the lytic cycle, the lysogenic cycle reproduces the viral genetic material without destroying the host. The lysogenic cycle of phage lambda begins when a phage binds to the surface of a host bacterium. The phage injects its DNA into the host cell, leaving the empty protein coat outside. The viral DNA is incorporated into the host cell DNA, where it is called a prophage. Every time the host bacterium reproduces, it replicates the phage DNA along with its own and passes the copies on to daughter cells. Occasionally, the phage DNA exits the bacterial chromosome and initiates a lytic cycle. The viral DNA takes over the metabolic machinery of the host cell to make phage DNA and proteins. The host cell lyses, releasing phages, which can infect other cells. This diagram summarizes the lysogenic and lytic cycles of phage lambda.
Okay, so bacteria aren't entirely defenseless against bacteriophages. Natural selection does favor some bacterial mutants that have surface proteins that are not recognized as receptors by certain bacteriophages. So if a bacteria has slightly different surface proteins that aren't recognized as receptors, then that bacteria would be immune to a phage infection. Also, sometimes when phage DNA enters a bacterial cell, in some cases that DNA can be identified as something foreign, and something called restriction enzymes within the bacteria will cut up the foreign DNA, and the bacteria will not be affected by it. So there's a few differences between bacteriophages and animal viruses that might be able to help keep these separate in your brain. So what we've been talking about were bacteriophages for basically the whole first part of this video. And they typically have DNA as their genetic material, and they don't have any viral envelope. So as you remember from those animations, they all had DNA, and they all just had a capsid with no membranous viral envelope. A lot of animal viruses have RNA as their genetic material, and they have this viral envelope that we're going to talk about in a second. The viral envelope can be actually really useful to the virus because they help the virus bind to specific receptor molecules on the host cell. And the way they work is by basically making the viral envelope using some materials from the host cell's plasma membrane. And that makes it much more easily recognizable to whatever cell it's trying to infect. Okay, so this is what that looks like. Here we have an animal virus up here. We have the RNA. Not all of them have RNA, but a lot of them have RNA. And remember, bacteriophages typically have DNA. So here's the genetic material, which is RNA. Here we have the capsid made of the capsomeres. But on the outside, we have this membranous viral envelope. And it's got these proteins on it, which basically it got from the last cell that was infected by this virus. So it attaches to the host cell using these proteins that it's accumulated. The host cell recognizes it as something that's familiar and allows, basically fuses with the membranous envelope. And once it does that, the capsid with the RNA is allowed into the host cell. Once that happens, the capsid disassembles. The viral RNA is now inside the host cell, and a similar kind of process happens where the DNA of the host cell stops working. There's several different ways that this can work, but in this case we have the RNA, viral RNA working as a template, which is making new RNA. mRNA over here, which is going to create proteins, viral proteins, remember, not host cell proteins. And the other thing that can happen is the viral genome can be copied, and it can be copied because in this case the virus may have additional enzymes which help the RNA be copied. So the host cell uses the enzymes that the virus had to copy the virus's genome. So here you have multiple copies of the viral RNA. Here you have other RNAs being turned into mRNA and creating the viral proteins. And so once you have the viral proteins and the new copies of the viral genome, they will assemble again. Now over here, it's talking about the mRNA being used within the host cell's ER to create these proteins that will be incorporated into the viral envelope. So the ER is making these proteins, and then it's going to send these vesicles out, which will fuse with the plasma membrane. And then when the capsid with the RNA inside it leaves, it's going to take with it some of these glycoproteins that were inside the ER. And now this new virus will have these proteins on the outside, which again will help it bind to the cell. This is showing you what type of structure the coronavirus has, because that is the virus that's causing us so much trouble right now. Coronavirus is just a category of viruses. So this one that's bothering us right now is a brand new coronavirus that's never infected humans before until now. But here you have some components of its structure. We have that it has a viral envelope. And here are some of the symptoms that it causes. Severe acute respiratory syndrome. So SARS is one example of a coronavirus. And MERS is another example of a coronavirus, but also the common cold is an example of a coronavirus. Okay, so I thought there was going to be another video demonstrating this animal virus infection, but there isn't. So hopefully you got all that you need from this. If not, make sure you go through the textbook because they have a good description of it as well. This glycoprotein stuff, you may want to review that a little bit more because I think I kind of muddled that a little bit. But that's the end of this video, and then there's just one more video summarizing up viruses as pathogens.